So let's talk about those three properties that result from hydrogen bonds in a little bit more detail: cohesion, adhesion, and surface tension. Cohesion means that water molecules like to stick to each other through hydrogen bonding, whereas adhesion means that water molecules like to stick to other molecules through hydrogen bonding. And so if you look at the pictures I have for you, cohesion is responsible for the water beating up. You see that on the leaf. It's also responsible for beating up on that spider web, but it also adheres to the protein in that spider web. So that image shows you both cohesion and adhesion. And then finally, surface tension results from hydrogen bonds resisting being stretched. So it creates a nice tight surface, and the image there is showing you a water strider that can actually walk on water. And if you've ever skipped rocks on a lake, you know this feature of water. And just again, reminding you that if you look at the diagram here, it's kind of reminding you why these things happen, that there are hydrogen bonds, and this water molecule is forming hydrogen bonds with 4 other water molecules. And that is kind of the maximum number that the water molecules can account for in hydrogen bonding. So moving on to topic 1.2, we have the elements of life. And all organisms must exchange matter with the environment to grow, reproduce, and maintain organization. Atoms and molecules from the environment are necessary to build new molecules. So we're going to focus on carbon, nitrogen, and phosphorus right now. But if you see, I've got tiles from the periodic table, and you may have learned this as CHNOPS: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. These are the 6 elements that make up 96% of living things. The other 4% is made up of some other elements such as chlorophyll and calcium and potassium, and so they are important, but they are way less prevalent than these 6. So carbon, and we're going to talk more about this, but carbon is used to build all of the biological molecules: carbohydrates, proteins, lipids, and nucleic acids. Carbon is used in storage compounds like starch and glycogen, and also in cell formation in all organisms. It forms the cell wall in the form of cellulose in plant cells. Nitrogen is used to build proteins and nucleic acids, and phosphorus is used to build nucleic acids and certain lipids, the phospholipids. More on all of this is coming up. So I want to introduce those terms that I told you should be familiar with, it just in case you forgot. And these are the terms monomer and polymer. Monomer is the subunit that serves as a building block of a polymer. So mono means one, poly means many. So a polymer is a larger molecule that's made up of many similar or identical monomers that are linked together by covalent bonds. And I'm going to give you examples in each of the groups of organic molecules. So the monomers of carbohydrates are monosaccharides, which are also known as simple sugars. And you're probably familiar with glucose, but there are other simple sugars that I'll point out in just a moment. And I'm showing you the ring structure of glucose. The monomers of proteins are amino acids. And I just have a generalized image of an amino acid there. We'll talk more about what those components are in just a minute. And finally, the monomers of nucleic acids are nucleotides. A nucleotide is composed of 3 parts: a phosphate group, which is why we need that phosphorus, a pentose sugar, and a nitrogenous base. And that nitrogenous base is why we need the nitrogen. So let's take a look at carbon. This flow chart kind of shows you why carbon compounds are so important to living things. They make up carbohydrates, which consist of sugars and starches, and as I mentioned, cellulose, which is a structural component made of carbon, hydrogen, and oxygen. They are often called hydrates of carbon. Quite literally, there's twice as much hydrogen as carbon and oxygen. We also have lipids. Lipids consist of fats, waxes, oils, and steroids, and what they all have in common is they are nonpolar, and they are composed of carbon, hydrogen, and oxygen, and in the case of phospholipids, phosphorus. Nucleic acids consist of nucleotides, which contain carbon, hydrogen, oxygen, and phosphorus, and proteins consist of amino acids, which contain carbon, hydrogen, oxygen, and nitrogen. So I want to talk a little bit about the two reactions that are really important in building and breaking down our macromolecules. And these two reactions are called hydrolysis and dehydration synthesis. And you probably, from those names, hydro and dehydration, understand that they have something to do with water. And you would be correct. Okay, so dehydration synthesis literally means the dehydration or removal of water. And the way this works, if you look at the monomers, you've got a monomer which has a hydroxyl or OH group indicated in blue, and you've got a second monomer which has a hydrogen that is showing you, it's an OH group, but the hydrogen is in blue. And so what's going to happen is that we're going to remove the water by combining the hydroxyl from one monomer and the hydrogen from another, and that forms water. And then the resulting monomers are linked together to form a polymer, and they are linked together by a covalent bond. Okay, I'm going to give you specific examples with macromolecules in a minute, but this is kind of a generalized view of dehydration synthesis. I should also tell you that dehydration synthesis is known as condensation. And the way I like to think about it is you're condensing those two monomers by removing water to create that polymer. The opposite reaction is called hydrolysis, which literally means using water to lyse or break apart a larger molecule into smaller molecules. So if we've got that polypeptide or polysaccharide or nucleotide that is linked together, if we add water, we hydrolyze it, we're going to break that bond, and we are going to split those monomers into two and re-add that water to each monomer. The hydroxyl group will go to the first one, and the hydrogen will go to the second one. So let's take a look at hydrolysis in actual biological molecules. So I've got a disaccharide, which is a 2-unit sugar. So it's composed of two monomers. And my example is sucrose, which is probably what you put in your coffee or tea. And it's cane sugar. So we want to break that down so that we can digest it better. By the way, disaccharides are kind of how the sugars travel around. They're the transport forms of sugar. And we want to break it down. It does require enzymes and energy. The enzyme in this case is called sucrase. We add water, we're going to hydrolyze it, and when we do that, we are going to break it down into two glucose monomers. Okay, in the case of sucrose, we would get one glucose and one fructose. In the case of maltose, which is another disaccharide, you would get two glucose monomers. Thing to remember. So let's take a look at some carbohydrates and see how their structure and function is derived from the way that they're assembled. So again, just to kind of give you a flow chart, over here we've got our group, which is the carbohydrates. They can be either simple or complex, and you might know this from your own diet. If you eat simple carbs, you're going to get readily available energy, and that comes from your monosaccharides, which are glucose, fructose, and galactose. And disaccharides are kind of the way that they're transported around your body or a plant body. It's how the organism kind of moves their sugars from one place to another. And there's maltose, lactose, and sucrose. And you may be familiar with lactose. It's milk sugar, and you may be familiar with it because you've heard of something called lactose intolerance. And that means that individuals who are lactose intolerant do not have a functional enzyme lactase to break it down using hydrolysis. So more on that another time. I'm sure that will come up again. So if we look at the image where I've got starch versus cellulose, right? Starch versus cellulose is all in how those monomers are assembled. And what I would like you to see in starch, that is a storage form or a complex polysaccharide. Plant starches are a little bit different than animal starches. Glycogen is an animal starch. And in this case, when when this flowchart is talking about fibers, we're mainly talking about cellulose, which is the bottom image. So I'm sure you can notice that there's a difference between starch and cellulose. And if you look kind of, I'm going to draw an imaginary plane across these ring structures. The bond is below that plane. And it turns out that when we link our glucose monomers together using dehydration synthesis, if that bond occurs below that plane, we're linking alpha glucose monomers, and we can easily digest those. We have the enzyme that can break those bonds and release energy, and so we can use that for an energy providing molecule. If you look down here at cellulose, if you look at that imaginary plane, the bond is above the plane. And the reason, and the way I remember that is these are beta glucose monomers that are linked together. And I remember this as beta above. And it turns out that humans do not have the enzyme that recognizes that what we call a 1,4 beta glucose link. And the reason that we can't digest cellulose is we don't have that enzyme. So cellulose makes up the bulk of our waste products because we can't digest it. Of course, there are organisms that can. All you need to do is think about termites, and they can get their energy from eating wood. So let's take a look at one more example of the carbs and their sugar monomers. In this case, we're showing you another dehydration synthesis or condensation reaction. In this case, we have two monosaccharides, glucose and glucose, and we're going to build the disaccharide maltose. We're going to do that by removing a water molecule. When we form this, this is that 1,4 link, and this is called a glycosidic linkage. Remember that this reaction, dehydration, because we're removing water, requires energy and the correct enzymes to build that disaccharide, and then you would just keep doing that to build your polysaccharide. I want to talk about the last group of biological molecules that I'm going to get into detail about, and those are the lipids. Again, let's start with this flow chart here. So lipids are all grouped together because they are nonpolar, and you've got fats, sterols, and phospholipids. Fats are composed of triglycerides, and I'm going to show you how you can build a triglyceride using dehydration synthesis. Triglycerides are stored fat or consumed fat. They make up cell membranes. Specifically, phospholipids are important in cell membranes, and I'm going to go into detail about that. They're also extremely important in creating the myelin sheaths that cover your nerve axons. Sterols are very important. Sterols are steroid molecules which are characterized by being nonpolar, which is why they're classified with the lipids, and they are 4 linked carbon rings. Probably the most familiar one you know is cholesterol, and cholesterol is important in cell membrane structure. It helps to maintain the fluidity and provide structure for that cell membrane. We tend to think of it in dietary terms, and a lot of people think it is bad. And while it is, in fact, there is one type of cholesterol that is not good for you in terms of your blood vessels. Your blood is polar, and these cholesterol molecules are nonpolar, and they tend to get stuck to the inner lining of your blood vessels, and that can cause problems. But cholesterol is a really important molecule. It is the basis of your sex hormones, testosterone and progesterone and estrogen. It's also the basis for vitamin D. And then finally, phospholipids. Phospholipids are super important as part of every lipid bilayer, which makes up cell membranes. So that's kind of the breakdown of the lipid flow chart. They are nonpolar, and that's an important thing to remember as to why they're linked together. There are 2 things I want you to know. Differences in saturation determine the structure and function of lipids. So you've probably heard in terms of diet that there are saturated fatty acids and unsaturated fatty acids. Saturated fatty acids have no double bonds. They tend to be solid at room temperature, and they're more animal fats. In terms of your diet, they're less healthy for you. The unsaturated fatty acids do have double bonds. You can see that that is indicated by the green portion of that molecule, which means that they're a little bit more flexible. You can also have polyunsaturated fatty acids, which have more than one double bond. These tend to be your plant fats and or oils. They tend to be liquid at room temperature. And then finally, phospholipids. Phospholipids are really important molecules because they contain polar regions due to that phosphate. You can look up here. The phosphate is shown in blue, and it's hydrophilic, meaning that it likes to be near water. Hydro means water, philic means affinity for. But it also, because of the nonpolar fatty acid chains, is hydrophobic at the same time. And this makes it a useful molecule in creating that barrier as part of the cell membrane. So I've showed you this cell membrane. It's not a great image, but you can see that this is the phospholipid bilayer. The phosphate heads are at the top, and they point both into the cytoplasm and out to the surface of the cell membrane. And then we've got all of these fatty acid tails, which are pointing toward each other because they're hydrophobic. One other thing, you can see a cholesterol molecule is embedded here, and that gives it a little bit of fluidity. You can see one here, it's not labeled, but that's also a cholesterol molecule. And then one other key component, as long as we're talking about cell membranes, is proteins. And proteins can span the entire cell membrane, or they can be on the surface. And those are called peripheral membrane proteins versus the integral membrane proteins that you see there. Okay, so let's talk about how you build a triglyceride. So I'm showing you a fatty acid. In this case, it's palmitic acid, and you can see that this is a saturated fatty acid because there's no double bonds. You can also see that it's got this carboxyl group at the end, and it's going to give up a hydroxyl group. And then we've got a glycerol molecule, which is a 3-carbon alcohol, and it's going to give up a hydrogen when we use dehydration synthesis. To build a triglyceride, you need 3 fatty acids. So what's going to happen is we're going to do that, and we're actually going to remove 3 water molecules. So you can see that that is a triglyceride. You've got the 1 glycerol molecule, and what happens is this bond here is called an ester link. So where the water was removed in the dehydration synthesis, you get that bond, which is called an ester link, and that is an example of a triglyceride.